I'd like to start by thanking the British Psychological Society for sponsoring this session. In this talk, I will consider some of the neurocognitive evidence addressing the effects of poverty on education, particularly focusing on executive function skills. Meta-analyses have consistently shown an association between the socioeconomic status of a children's families and their educational achievement. Although effect sizes vary, a similar pattern is found across multiple countries. One meta-analysis of US studies suggested a small to medium effect size of 0.27. The Commission on Inequality in Education in the UK published a report arguing that this performance gap was not narrowing, and there is evidence that gaps are being widened by the pandemic. However, the mechanisms of transmission from parents' SES to children's education are not yet well understood. There are a range of candidate influences. Economic hardship may influence the child's early brain development due to nutrition, stress, or other effects on the mother. It may influence the child's level of cognitive stimulation. Cognitively, the strongest effects of SES are seen on language development and executive function skills. It may influence other aspects of the child's psychology, such as their self-concept. Economic hardship may produce negative effects on the parent's emotional state, their relationship, and their parenting behavior, impacting the child. Children may have access to fewer physical, cognitive, and social resources. The values and expectations in families with respect to education may differ. There may be influences stemming from the neighborhood and schooling in deprived areas. And children's behavioral adaptations to sometimes more dangerous and impoverished environments, including vigilance or a scarcity mindset, may ill-suit them for the classroom. Lastly, deprived environments may be associated with chronic stress and a greater risk of poor health. A neuroscience perspective on poverty in education has to consider the nested influences on the child's educational outcome. Proximal causes affecting development and brain function are child factors, but these are nested within constraints operating at the school level, such as teacher skill, society and family factors, such as cultural influences, and government factors, such as the education budget. Attempts to close educational gaps must address all of these constraints on children's success. In this talk, I'll briefly consider two neurocognitive aspects of this relationship: executive functions, sometimes referred to as cognitive control or behavioral regulation, and the proximal effects of differences in SES on brain development linked to cognition. SES and executive functions are independent predictors of children's academic success and other important life outcomes, such as employment, but they are also correlated with each other. One study by Hackman et al. Followed a cohort of children as they started school. Differences in executive function skills associated with SES were already present before the children started school and did not narrow across middle childhood. One might imagine schools would serve to equalize children from different backgrounds, but this result suggests the effects of home environment retained a more powerful influence than the school. The study also implicated childhood home characteristics, including maternal sensitivity, in producing differences, perhaps related to the caregiver's scaffolding of young children's development of behavioral regulation. One challenge in investigating the relationship between SES and executive functions is that both are complex constructs, each measurable in a variety of ways. Moreover, some view executive functions as tightly linked to overall cognitive ability or IQ. Is there any specificity of the effects here? I'll briefly present some results exploring this question from a UK cohort of early adolescents collected as part of the SCAMP study investigating mobile phone usage, carried out with colleagues at Imperial College. Here we see the range of measures that one might use to assess socioeconomic status. This is not completely inclusive. These include parental education, parental occupation, index of neighborhood deprivation, and whether children are attending a state or independent school. In addition, we've broken down executive functions into a range of tests considering switching, working memory, planning, and behavioral regulation. Separately, we also assess nonverbal fluid intelligence. What pattern of relationships did we see? This analysis was led by Lizzie Booth and Iroaz Dumantai at Birkbeck, looking at the results of regression models linking SES metrics to individual executive function skills, having control for age, gender, ethnicity, and English as a first language. The numbers vary per measure, but the smallest number of participants in each of these regression models is 900 individuals. And the average age of these children, drawn from the London area, is 12 years of age. You can see in the colored cells that there are specific relationships between different SES metrics and different executive function measures. School status seems a fairly robust marker. Notably, Several of these effects remain significant having control for nonverbal intelligence, implying that SES differentially and specifically impacts executive function skills. These kind of associations prompt an investigation of proximal cause. We now move on to the results from brain imaging studies. As we've heard from Sebastian Lepina's talk, there's been an increasing literature examining differences in children's brain structure and function associated with differences in SES. Reviews of this literature, such as that by Martha Farah, have suggested that while many differences have emerged, few are consistent or specific. On the whole, effects on brain structure seem a better indicator of accumulated environmental effect compared to function, since these are not influenced by task demands. That is, children from different backgrounds may respond differently when asked to perform cognitive tasks in a brain scanner. Two of the notable findings point to different proximal causes. The findings by Kimberly Noble and colleagues in a US sample suggest a small nonlinear association in cortical surface area linked to family income, and effects strongest at lowest levels of income. This association was strongest in temporal and frontal regions and mediated performance on cognitive tests. It is consistent with the behavioral differences observed in language and executive function skills, respectively. The regional pattern points to experience-dependent effects and therefore the impact of differences in cognitive stimulation. By contrast, the data from Betancourt indicates differences in gray matter in US female African-American infants of 1 month of age, and these results are more consistent with effects in early brain development. One challenge of finding brain differences associated with poverty is the message taken from them by policymakers. In a sense, it is a double-edged sword. The presence of differences in brain structure is a powerful demonstration that the effects of poverty on development are real, yet they may encourage policymakers to think that there is little that can be done to change outcomes. To derive implications of observed brain differences for the opportunity to intervene, we need mechanistic models which specify proximal causes. For example, whether a child's father went to university or not is not a proximal cause, but how that alters the type of parenting or level of stimulation in the family environment is. One way to proceed is through the use of computational models, which will allow for testing the effects of interventions. Our challenge here, and indeed theoretically, is that accounts of SES effects have to be embedded in wider theories of the development of brain and behavior, and integrated with findings that differences in brain and behavior are often highly heritable, that is, due to genetic variation. For example, indices such as behavioral accuracy, cortical thickness, and cortical surface area show developmental trajectories. Indeed, cortical thickness reduces with age due to pruning and optimization of neural circuits. SES effects instead examine cross-sections within this picture. I will briefly describe a computational model employing artificial neural networks, which attempts to bring these data sets together and to test the effects of interventions. An artificial neural network abstracts and simplifies key properties of real neural systems taken to be important for information processing. This particular model implements its processes of growth and pruning to capture early resources for plasticity, activity-dependent strengthening of connectivity, and later pruning for optimization. The model contrasted 3 hypotheses for causal pathways through which SES effects operate. Postnatal cognitive stimulation, prenatal brain development, or both operating in a correlated fashion. You can see in these graphs that all 3 model conditions capture the broad developmental trajectories. Here, number of connections in the network was used as an analog for cortical thickness, and total connection magnitude as an analog for cortical surface area. In these models, genetic variation influenced variation in how many of the low-level neurocomputational parameters. Genetic variation influenced variation in many of the low-level neurocomputational parameters, capturing the high heritability of brain structure and behavior. Turning to SES effects, the cognitive stimulation condition gave the best fit to the Noble et al. Data. But what's of more interest here is that with the model in hand, we can start to run the kinds of experiments that nature and ethics prevent us from doing. We can simulate the same child raised in different levels of impoverishment. In these data, we carry out an early intervention, maximizing and equalizing the cognitive stimulation for all children at an early point in development, say around age 5. You're looking at how this changes the population distribution of behavior and brain structure later in childhood. Gray showing the control condition, yellow showing the effects of intervention. You'll see that all conditions showed a strong behavioral response to intervention, with relatively subtle effects on brain structure. Notably, the cognitive stimulation and network growth conditions produced exactly the same mean improvement, but the response was lower in the correlated effects condition. However, the conditions predicted a different pattern of response across SES levels, with stronger behavioral effects for low SES kids, where the causes were primarily from differences in cognitive stimulation. These kinds of interventions targeting cognitive stimulation are being developed and tested, both for executive function skills and for language development. The mechanistic model, which situated SES effects within a developmental framework and against a background of highly heritable individual differences, suggested two findings. First, the presence of brain structure differences linked to SES does not militate against behavioral interventions. Second, cumulative risks, here the combination of effects on cognitive stimulation and brain growth, produce the poorest outcomes. In conclusion, there are multiple contributions to the impact of poverty on educational outcomes. Cognitive skills may provide possible mediating factors and targets for intervention, particularly through targeting the home environment, which has stronger and earlier effects than the school environment. However, returning to the nested constraints that shape educational outcomes, social and educational factors may also support resilience. Brain-level data offer a persuasive contribution to the debate, but causal models are required to derive the implications for policy and to avoid the risk of miscommunication. Lastly, looking forward, theoretical challenges are emerging in how we interpret increasing evidence of gene-environment correlations in predicting educational outcomes. That is, polygenic scores derived from DNA predict variance in academic achievement. SES predicts variance in academic achievement, but these two predictors are to some extent correlated with each other. What are the implications for interventions and for policy? In addition, much of the research on poverty has been derived from the lowest quartiles of children in high-income countries. However, there is evidence that findings such as those on executive function skills cannot necessarily be generalized to SES effects in low and middle-income countries. Therefore, the generalizability of the scientific evidence for the issue of global poverty needs to be carefully considered. Thank you. I'd like to thank my collaborators and funders, and thank you for your attention.